Hello everyone, welcome back to another video. In this video, we are going to talk about the life cycle of Trypanosoma gambiense, which is the causative agent of sleeping sickness. In this particular video, we are going to look at the life cycle of this parasite and how it is responsible for one of the most deadliest diseases, that is human African trypanosomiasis or sleeping sickness. Trypanosomes are a group of blood and tissue dwelling unicellular parasites. They belong to the category of zooflagellates. That means that they are flagellum bearing animal like organisms. And they are said to be parasitic in a variety of different vertebrate organisms, including human beings, and invertebrate organisms like insects. Trypanosoma word is itself derived from two Greek words, trypanes, which means to bore, and soma, which means body, because of their characteristic corkscrew like motion. Now, these trypanosomes do not have chlorophyll bearing chloroplasts as well as paramyelin granules. So, they are said to be showing heterotrophic mode of nutrition and more specifically parasitic mode of nutrition. They are also categorized by the possession of a flagellum, which is arising near the posterior end of the body and it extends towards the anterior end of the body, as we are going to look in just a minute. Now, even though there are many trypanosomes, two of them are very important for us because they are pathogenic in man. The first one is Trypanosoma brucei and the other one is Trypanosoma cruzi. Trypanosoma brucei is said to cause human African trypanosomiasis or sleeping sickness, whereas Trypanosoma cruzi causes Chagas disease in Americas. The Trypanosoma brucei itself contains of three subspecies which are all pathogenic. Trypanosoma brucei is a pathogenic species, however, it is not pathogenic in human, it is pathogenic in cattle. Trypanosoma brucei gambiense is said to be causing West African sleeping sickness, whereas Trypanosoma brucei rhodesiense causes East African sleeping sickness. In this particular video, our target would be Trypanosoma gambiense, which is responsible for causing West African sleeping sickness. In this picture, you can see the image of Trypanosoma in a blood smear of an infected individual. Now let's look at its taxonomic position. Its phylum is Sarcomastigophora, subphylum is Mastigophora, class is Zoomastigophora, genus Trypanosoma, species Brucei, and subspecies Gambiense. Just like in this picture also, you can see that there is a flagellum at the posterior end. This is the posterior end and this flagellum is extending towards the anterior end. And by the way, this taxonomic position has been taken from 5th edition of Barnes. Now the trypanosomes are said to be existing in multiple polymorphic forms. And there are 4 important polymorphic forms that have been recognized. Let us look at them one by one. The first form is amastigote form. In this form, the organism is round or oval. There is a nucleus present along with the blepharoplast and kinetoplast. Blepharoplast or basal body is the structure from which the flagellum is going to arise, whereas kinetoplast is a mitochondrial structure which has DNA. Now in amastigote, there is no flagellum. Basically, flagellum is reduced and it remains embedded in the cytoplasm. So even though it has not been shown in this particular picture, there is a flagellum which remains embedded inside the cytoplasm, but the flagellum does not appear outside. Next, you have the promastigote form, or also known as the leptomonad. This form is elongated, has a large nucleus, and the blepharoplast and kinetoplast are located anteriorly. As you can see over here, they are located anteriorly. This form has a short and free flagellum outside the body. Then you have the third form, which is epimastigote, also known as the crithedial form. In this form, the body is elongated However, the blepharoplast and kinetoplast are located immediately anterior to nucleus. And now there is a small and poorly developed undulating membrane. Now you might wonder, what is this undulating membrane? Actually, the body of the organism is covered with a structure which is known as pellicle. And when the flagellum beats, the pellicle is thrown into a membranous fold which is known as the undulating membrane, as you can see over here. This is the undulating membrane. Now, in this undulating membrane, what is happening is that you can see the flagellum being arising, and it is actually extending as a tip of the undulating membrane, and at the anterior end, it is emerging as a free flagellum. The fourth form is the trypomastigode form or the trypanosomid form, which is also elongated. However, here the blepharoplast and kinetoplast are located at the immediate posterior end, and the undulating membrane is well developed here. In the previous case, the undulating membrane was very inconspicuous, it was very poorly developed, but in this particular form, in trypomastigode, the undulating membrane is very well developed, and the flagellum arises at the posterior end from the blepharoplast, goes as the tip of the undulating membrane, and at the anterior end, it arises as a free flagellum. The trypamastigode form is then further said to be existing in three forms. You have the long and slender form, in which you can see that there is a free flagellum. Then the immediate form, in which the flagellum is somewhat shorter, and the short and stumpy form, in which there is no free flagellum present outside the body, and the flagellum is rather inside the body only. Now let's talk about its distribution. The Trypanosoma brucei gambiense is endemic in Western and Central Africa. The maximum cases of sleeping sickness are reported in the Democratic Republic of Congo. There were around 600 cases reported in 2022. Trypanosoma brucei rhodesiense, on the other hand, is restricted to Eastern and Southern Africa. Now let us talk about its morphology. As we have already seen, the parasite has an elongated body and its posterior end is blunt, whereas its anterior end is pointed. In the human host, there is the long, slender, short, stumpy or intermediate form found. So basically the trypamastigode form is found. And in the CC fly, which is its invertebrate host, the long, slender form is found in the midgut, whereas the short, stumpy form is found in salivary glands. Also, the epimastigode form is found in the salivary glands. We are going to talk about this in just a minute, so it will become more clear to you at that time. The whole body of the parasite is covered with a thin pellicle, which is supported by fine microtubules from underneath, and there is a single flagellum which is arising from the basal body or blepharoplast, which is located at the posterior end. The flagellum runs forward and it becomes free at the anterior end. When the flagellum beats, the attached pellicle is pulled up into a membranous fold, which is called the undulating membrane, as we have already discussed. Inside the cytoplasm, there is a nucleus, kinetoplast, Golgi, endoplasmic reticulum, vacuoles, and ribosomes which are present. Now let us talk about the habit and habitat of this parasite. The parasite in itself is diagenetic, meaning that it has two hosts. It requires two hosts to complete its life cycle. The primary host of the parasite is a vertebrate, like human beings, and it mostly lives in the bloodstream and lymph nodes of human host during the early stages of infection. And during later stages of infection, the parasite invades the central nervous system and it invades the cerebrospinal fluid, which actually manifests into sleeping sickness. The secondary host of the parasite is the one which is the CC fly, which is also its vector, which is responsible for transmission of the parasite from one infected human being to another healthy human being. The CC fly's biological or scientific name is Glossina palpalis. The parasite also has reservoir hosts. Recall that a reservoir host is one in which the parasite stays without any undergoing any developmental changes until the time that the parasite is taken up by the CC fly and injected into another potential host. The reservoir hosts for this parasite include domestic and wild animals like monkeys, pigs, dogs, etc. Now let us talk about the actual life cycle in man. To a healthy human being, the infection occurs by the bite of a CC fly, which carries the infective metacyclic form of the parasite in its salivary gland. This infective metacyclic form is actually the short, stumpy trypomastigote form that we have just seen. Now, this metacyclic form will become transformed into a long, slender form, and these long, slender forms are able to swim freely with the help of their flagellum. And they also multiply by the help of longitudinal binary fission. Now, initially, they are utilizing glycolysis for energy deriving, but then once antibodies are formed against the parasite, they are no longer able to derive energy from the glycolysis pathway, and then they shrink into this short stumpy form, and during this transformation, intermediate forms also appear. Now, the short stumpy forms do not feed. So, if they're not picked up by another CC fly, then they're going to ultimately die in the human host itself. Now, what happens when the parasite is picked up by the CC fly in its blood meal? The CC fly is taking the short stumpy form of the parasite in its blood meal, and once it is in the midgut of the insect, this parasite will develop into the long slender form once again, and there also it is going to divide by longitudinal binary fission. The parasite then will make its way into the salivary glands via esophagus and mouth parts of the insect, and inside the salivary gland lumen, the parasite is going to transform into the crithedial form. So, if you recall, the crithedial form is the epimastigote form. Then this crithedial form again multiplies and then finally transforms into the slender metacyclic forms. These slender metacyclic forms are going to be entering into a healthy human being when this CC fly is going to bite that human being. Now let us have a look at the life cycle as a whole. So in the first step, as you can see that the CC fly is taking a blood meal from a healthy human being, and this CC fly, which was carrying the metacyclic trypomastigotes in its salivary glands, when it is taking the blood meal, it can pierce the skin and it can inject these trypomastigotes inside the bloodstream of the organism, that is human being. Now the trypomastigotes remain extracellular. That means that they do not enter the blood cells, rather they are developing inside the plasma itself. Inside the plasma, if you remember, these metacyclic trypomastigotes are going to develop into the long slender form, and then the long slender form is going to divide and then get converted into the short stumpy form. So here also you can see that it is written injected metacyclic trypomastigotes transform into the bloodstream trypomastigotes, and they are carried to the other sides, that is the lymph nodes as well as the the cerebrospinal fluid. The trypomastigotes are dividing by the longitudinal binary fission. That means that their body is going to split into two halves across the longitudinal axis. So if this is my organism, the body is going to split into two halves across the longitudinal axis. Trypomastigotes are then going to multiply in various body fluids, that is your blood, spinal fluid, lymph, and these circulating trypomastigotes are then undetectable, the short stumpy ones, and they are actually the ones which are picked by the CC fly during its blood meal. Now inside the gut of the CC fly, they are going to again transform and they are going to lead into procyclic trypomastigotes, which are going to again multiply by the binary fission. They enter your they They enter the salivary glands and transform into epimastigotes, which are then multiplying in the salivary glands and resulting in the formation of the metacyclic trypomastigotes, which are going to be the infective stages for the parasite. Infective for human being. Now let us talk about the disease which is caused as a manifestation of this infection, that is sleeping sickness. Now as the name suggests, sleeping sickness, in this particular situation, the person in the later stages of the disease is induced in a rather sleeping like state. Person likes to sleep a lot and it ultimately ends up in a coma like position. So the symptoms include the initial inflammation of the area where the CC fly has bitten the person. So, multiplication of the trypomastigotes at the inoculation site, that is where the CC fly actually injected the trypomastigotes, is going to produce a localized inflammatory lesion. The invasion of the lymphatic glands also causes them to swell, and later, as the parasite is invading the cerebrospinal fluid, this actually leads to manifestation of the disease. The patient is said to experience recurrent fever, weakness, weight loss, anemia. There is an increase in the pulse rate and a severe headache. Patient falls asleep first at regular intervals and then a coma-like state is induced, which is followed ultimately by death. Now, in order to diagnose this disease, there are three steps which are involved. The first step is screening for potential infection. So, this is done using serological, that is serum based tests, and they are only available currently for Trypanosoma brucei gambiense. Then we confirm the parasite's presence by observing it microscopically. Then you have the staging of the disease progression via clinical examination and analysis of cerebrospinal fluid obtained by lumbar puncture if needed. So basically the first step, you're going to use basic tests, serum based tests, so as to uh, screen whether the person has the chance of carrying the parasite or not. Then if the screening test it comes out to be positive, then the next step is actually looking at the blood smear under the microscope to test for the actual parasite and its presence in the blood plasma. And then if the parasite is found in the blood plasma, the next step is to see how much the disease has progressed. That is what we do. For that, what we do is that we take out the CSF by lumbar puncture, most probably, as it has been shown in the above picture. Also here the lumbar puncture has been shown. So this is what we will be doing for extracting the CSF, and then inside the CSF we are going to see whether the parasites are present there or not. Because if the parasites are present in the CSF, this would indicate that the disease has progressed to later stages and it is much more difficult to cure, or rather incurable, during later stages of infection. So early diagnosis is very important because the later stages are neurological, and in that particular stage the chances of recovery are extremely less. Now the ways to treat it will depend upon the form of the disease and also the stage. As we said that the earlier it is detected, the more chances of it being able to be treated are there. Treatment in the second stage would require drugs that are crossing the blood brain barrier because now the parasite has entered your central nervous system. The ways to prevent the disease, that is, prophylaxis of the disease, is obviously by eradicating or killing the vector which is responsible for its transmission, that is CC fly, and that we can do by destroying its habitat, that is, clearing out the bushes and the low trees along rivers in the endemic areas, and also we can spray insecticides like DDT, which can kill the insect. So this was our video on parasite Trypanosoma gambiense and how it causes sleeping sickness. Thank you for watching.